Every 3 seconds, someone is diagnosed with Alzheimer's disease. They're told that the reason they've been forgetting to turn off the stove and having difficulty matching names with familiar faces is because of a buildup of plaque inside their brain. They're told it's a fatal disease. And when they ask what can be done, they'll be given a prescription for a drug that may make them feel better for a little while, but will do nothing to prevent their inevitable decline. 1 in 3 people over the age of 65 will die with dementia. They won't remember their vacations to Europe, their spouse of 40 years, or recognize their children. And there's nothing that we can do about it. Currently, 50 million people are living with this reality. And by 2050, that number is going to nearly triple to 130 million people. I was in pharmacy school when I learned this, and I was floored. How can this possibly be the best that we can do for these patients? Tens of millions of people suffering one of the worst fates imaginable, slowly drifting into solitude, forgetting everything they've ever known or loved. And that's when I decided to shift my research focus to Alzheimer's disease and pursue a PhD after I graduated. The goal of my thesis is to dramatically change the outlook for patients diagnosed with Alzheimer's disease by developing a drug that can protect nerve cells in the brain called neurons from deadly amyloid beta. Alzheimer's disease, in the most general sense, is the result of a dying brain poisoned by amyloid beta. Connections between neurons that make up the physical basis of our memories are lost, and over time, the cells die and the brain shrinks. Our lab has spent nearly a decade studying how amyloid beta makes the brain sick, and we discovered a protein on the surface of neurons that binds really tightly to amyloid beta and sets off a catastrophic cascade of events inside neurons. Last winter, I finally discovered a drug that can completely block amyloid beta from binding to neurons, and that's what I'm showing you here. In green, I'm showing you where the neurons are, and in red, I'm showing you the amyloid beta. In the first column, I didn't add any amyloid beta. In the second, I've added amyloid beta, and you can see that it's binding everywhere that the neuron is. In the third column, I've added the same amount of amyloid beta, but I've also added our drug, and it's completely blocked amyloid beta from binding to the neurons. Next, I designed an experiment to test if my drug can improve memory. So I gave my drug to mice that express the same mutant genes that cause Alzheimer's disease in humans. And after a month of treatment, mice that previously couldn't remember where a hidden platform was located could find that platform just as quickly as their non-mutant siblings. We're really excited about this new treatment strategy for Alzheimer's disease. Protecting neurons from toxic amyloid beta means that if a person started taking our drug when they began having symptoms, the neurons would have a chance to get healthy again, as if the amyloid beta isn't even there. I know there's a long road ahead before we know if our drug works in humans, but the potential to change the future for 130 million patients and their families is incredibly exciting. Thank you for your time.